Hi, I'm Yanagisawa, CEO of CellFiber. We utilize the CellFiber to make cell and gene therapy affordable for all. Today, the cell therapy is becoming a reality, but there is a significant obstacle to widespread cell therapy, which is poor manufacturability. Cell and gene therapy is still very expensive and can lead to limited number of people and the cost rates remain very high. Why? So manufacturing for cell and gene therapy is a challenge. So there are the mainstream. The 2D cell culture is well established, but not scalable. Another one is 3D cell culture, which is potentially scalable, but people often suffer from reduced the quality. You may wonder why cells are so unhappy during 3D culture. With the conventional cell culture technology, the microenvironmental condition to the cells is not consistent across depending on the culture scale, which means when the bioreactor becomes larger, you need to more vigorously stir the system to keep the cell culture media homogeneous. But when you mix the system vigorously, the more shear stress are applied to the cells, and which leads to the cell damage and even cell death. So our solution is can provide the consistent cell culture condition regardless of the scale. So here is our solution, CellFiber. What you see here is the hair thin like hollow gel fiber, and you can incorporate cells inside. This hollow gel tube serves as a mechanical barrier against the shear stress, and then you can maintain the consistent microenvironment regardless of the culture scale. What you see here is the real-time video for the fiber formation. You need to use the 3 fluid cell suspension and gel precursor solution and the gelating solution. When three fluid mix together, gelation happens and fibers are formed. So this process is very quick, and also when necessary, you can remove this hydrogel tube by simply exposing some chelating reagent. Here I'm showing the application for the iPSC expansion. We have already successfully established 20 billion iPSC expansion in 2-liter bag, where the cell density is 10 to the 7 cells per milliliter, which is 10 times higher than the conventional culture method. The good point is you can easily conduct the perfusion culture thanks to the cells are always separated by the hydrogel tube. The automation and in the closed system is possible, and then you can reduce the labor work. Here I'm showing how cell growth happens inside a fiber. You usually start with the diluted condition, then within a week, you can have the 50-fold or 100-fold the expanded cells like this. And importantly, even at a large scale, the harvesting processes can be done very quickly, as you can see here. Also, the improved yield, but you can maintain the quality, as you can see here. Those are the surface marker, typical surface marker for the iPSCs. And these this 2D one is the fiber, and the 2D is a conventional one. And this technology is competitive. The multi-tray, the batch scale is limited, and you can say same thing for the hollow fiber. And stirred tank, as I mentioned in the beginning part, difficult to maintain the quality. Our solution can satisfy all the points customer required. Thanks to the characteristics, we can provide this benefit to the customer tenfold, the 10+fold products, and also reduced fixed labor cost. We have already have the 15 plus the collaborator, including the US-based CDMO and licensed to the 2 company, and raised this money. And we also have the academic partners like this. And here is our team. We are capable to develop our product solution affordable in the market. So if you are interested in our technology, we are very happy to talk with you. This is the end of my presentation. Thanks for your listening.